Welcome everybody to the GlycoNet ACS Joint Series webinar. I am very pleased today to be able to introduce Dr. Wade Abbott, who's a research scientist at the Agriculture and Agri-Food Canada. Dr. Abbott received his bachelor's in biology from Trinity Western University in 1998 and his PhD in biochemistry under the supervision of Professor John Alcio at the University of Victoria in 2005. He did then a postdoctoral research fellowship with Professor Alistair Boriston, who is very well known for his work in carbohydrate interactions, and especially in enzymes, followed by a second postdoctoral fellowship with Professor Harry Gilbert at the University of Georgia's Complex Carbohydrate Research Center in metabolic pathways and intestinal bacteria, which obviously has informed quite a bit of Dr. Abbott's work. In 2011, Dr. Abbott began his independent research career with Agriculture and Agri-Foods Canada in Lethbridge as a research scientist, and his group research themes include the discovery of carbohydrate active enzymes, metabolic pathways, agricultural glycomics, and carbohydrate-based innovations for the production of food animals. And I'm looking forward to his talk entitled Glyco-enabling Tools for Study of Microbial Glycan Metabolism in Agriculture. So take it away, Wade.
So thank you for that very kind introduction, and I'm looking forward to this chance to present some of our current projects where we're taking current approaches to study contemporary problems in agriculture. So we can think of agricultural systems as complex ecosystems involved in the generation, modification, and consumption of glycans. And there are many opportunities to improve primary production, bioconversion, and clean technologies for carbon sequestration, animal, plant health, and ultimately ushering in a new era for sustainable agriculture and a greener economy. So this is a figure we're putting together for an upcoming review, where we're looking at a few different systems within potential farms. And if we consider these different interactions, we can look at the complex carbohydrates that are present, excuse me, in these different systems. So first of all, we can consider plant cell walls and primary production, feed conversion, so how these crops might be consumed, and overall plant health. We can also consider soil or plant exudates. So these are basically plant mucins that are secreted into the soil and regulate soil microbiomes. We can consider host glycans and omucins. So these are involved in innate immunity, regulation of microbial communities at mucosal surfaces. Also the production of milk oligosaccharides. So here we're looking for ways to improve neonate health and optimize artificial formulas for livestock. A current emphasis of agriculture is also looking for climate solutions. So we can look at what's going on in the soil: carbon sequestration, soil carbohydrates, polysaccharide turnover, and deposition mechanisms. Also, we consider horticulture and the production of fruit. So pectins are quite important here, and looking at their modification and their role in plant health and the prevention of infection by soil and fungal pathogens. And finally, our group is also interested in the production of complex carbohydrates in different ecosystems, such as the marine environment, and their application back into agriculture, whether that relates to reduction in greenhouse gas emissions, improved food safety, or even biofertilization. So a couple examples from these different systems include some work that we've actually commenced with Michael Hahn at the CCRC, where we're using his Glycome Profiling array. And so in this study, what we wanted to do is look at the glycans that are present in barley straw and compare them with what's present in the total tract indigestible residue, or the feces. And so by comparing these two different samples, we actually get an idea of the overall changes in glycan composition that's going on during digestion. And in particular, we can look at the accumulation of particular glycans, which are indicated here with the white boxes. And these represent potential targets for improving digestibility with the addition of new enzymes or even plant cell wall engineering. Also, we have some current research. We have a project funded by GlycoNet currently to look at the infectious disease and the role of host glycans in prevention. So for example, necrotic enteritis is also considered the billion-dollar disease. It's caused by Clostridium perfringens. And if we look at the genome of Clostridium perfringens, we can see that it is well equipped for the degradation of O-glycans. And so this is some work ongoing with Alberta, where we're looking at the structure-function relationship of enzymes involved in O-glycan modification. So for this talk, I'd like to focus on two different stories and how we can use our knowledge of glycans and glycan structure to regulate microbial abundance and function. So first of all, I just want to introduce the concept of PULS, or polysaccharide utilization loci. Now, these are pathways found in microorganisms involved in the metabolism of complex carbohydrates. And so you need a few different features here. You need the ability to transport glycans into the cell, and typically PULS consist of these Sus-like systems, which includes a Sus-C-like and a Sus-D-like protein. So this is the poran and a cap, and then also other surface glycan binding proteins, shown here in yellow, which help to recruit and concentrate glycans on the surface. A microorganism also needs the ability to sense the presence of glycans and respond, and enzymes involved in the recognition and modification, ultimately the depolymerization of these substrates for import. So this is from a paper that's going on 10 years now, we published with Eric Martens. And back then, this was quite informative, and actually this paradigm is held true over the last decade here. But we're looking at two related Bacteroides species. So Bacteroides theta iota omicron and Bacteroides ovatus. And you can see that they actually share some of these pathways, and these are shown in green. But each species has specialization in its metabolism as well. So in B theta, we can see here in orange that they're specialized for the consumption of O-glycans and pectins, whereas B ovatus is specialized for hemicelluloses. And so this we've actually discovered now holds true even to the strain level. And this places importance on characterizing different bacteria by whole genome sequencing. Now, PULS don't just exist in the gut. They exist in every ecosystem on the planet. And they are involved in the metabolism of the usual suspects of polysaccharides that you would find in that ecosystem. For example, the ocean, freshwater, forest soils, etc. So I just want to focus on first story here, where we looked at bacteria from the human gut. And more recently, we've discovered how it may impact the intestinal health of cattle. So this story was first published in 2015 in Nature, and it received a lot of popular press. I'm not sure they captured the true, the take-home message of the actual paper, but it was fun to interact with some of these different publishers. So the story here was that yeast mannan, which is a homopolymer of mannose, connected through different linkages, which are shown here in different colors, and they can be alpha-1, 2, alpha-1, 3, and alpha-1, 6, to form this highly complex structure that's presented on the surface of yeast. Now, B theta has pathways involved in the metabolism of this sugar. There are surface enzymes that are involved in the initial processing to generate complex fragments that are transported into the cell. And this is where the majority of the enzymes are located. And they work together to completely depolymerize the sugar into mannose. Now, this is called selfish metabolism. So most of the processing is happening within the cell. And this is to prevent substrate loss to the community. So why would we have bacteria adapted to the consumption of yeast? Well, it's been a component of the human diet for millennia. Of course, yeast is involved in leavening of bread and fermenting of food and beverages. But also yeast is a promising application for beef production. So in Canada and in other countries, there's a rising demand for beef for its citizens, but also for export. But the beef industry is facing several production challenges related to the phasing out of antibiotics and antimicrobials. So there's a real opportunity here for new innovations to improve cattle health and performance. And these include host-adaptive probiotics, prebiotics to improve feed efficiency. But there's a real lack of evidence-based solutions to study and tools to study these interactions, not just in livestock, but also in human health. So our approach was we wanted to isolate bacteria out of the rumen to develop some host-adapted probiotics. And so we started by taking yeast mannan as the sole carbon source, and we isolated using selective isolation bacteria from the rumen, and we called these B theta bovine strains. We isolated well over 100 these bacteria. But when we culture these in solution, we actually noticed a very interesting phenotype. These bacteria clustered into two groups. So we had a high grower, which we considered maximal growth at about 0.8, after 24 hours, and medium growers, which reached about half the density. And this was reproducible and heritable feature. So we sequenced the genomes and did comparative genomics on a number of these strains. And the thought was that we would, find a new pathway, or we could explain this genetically within the PULS why we have these two different growth phenotypes. Now, interestingly, when we look at B theta control strain, we see that it does have this first PUL here, and this PUL is only conserved in 2 of the genomes that we sequenced. However, these are medium growers, whereas B theta is actually a high grower. So we couldn't explain the addition of this single PUL responsible for this growth phenotype, and other medium growers didn't have this PUL. All the high growers were actually that we isolated were missing this first pathway. So the conservation of PUL abundance and structure cannot explain this growth phenotype. So we're curious if there's actually differences in the genes themselves that might be responsible for the efficiency or specificity of the enzymes. And so we subjected all the genes to our pipeline we call Saccharus, which is an automated tree generator for Kzyme. So it interacts directly with the KZ database, extracts sequences, and we can get rapid and accurate prediction of function by generating trees using enzyme characterized sequences. We can do this at the single sequence genome and metagenomic levels. So when we did this for the PULS, we actually found that there is complete conservation. Now, we're focusing here just on GH92 and GH76. These families are involved in the hydrolysis of alpha-1, 2, 1, 3, 1, 6 mannitol linkages. And so if we look at each of these individual enzymes, they actually are represented by these islands. So they have complete conservation within the different PULS. So you have absolute conservation of Kzyme sequences between the high growers and medium growers. We next turn to RNA sequence analysis of the MAN-PULS. We wanted to make sure that they are induced. We thought maybe there might be some new PULS present or some other features that we were missing. And indeed, we see that all the PULS are actually induced within each of the strains. We did find a new PUL in the high grower in B theta, which is also a high grower. We call this PUL 55. So we were quite excited by this. We thought maybe this could explain this differential growth phenotype. And when we look closer at PUL 55, we found in our isolate, there's actually the insertion of 2 new genes, a 92 and a 76. And so we're very curious about this. The 76 was predicted to be surface exposed. So maybe this would be unlocking this differential growth phenotype. We tested them against a variety of different substrates involving different yeast mutants. And here's the wild type substrate. So when we screened our 76 against these different substrates, we're very excited to see it was actually active on the wild type yeast mannan. And this is the first time a single endoactine enzyme had been shown to digest yeast mannan. So we carried on. We wanted to look at some of the products that were generated from these reactions. So we used 2 different linkage methods. This is a recyclization method. So we're looking for the presence of the furanose at the reducing end. And this was a pre-reduction method. So we're looking for the production of a mannitol at the reducing end. So in both cases, we saw that there was a 6 mannocell, which could be explained by cleavage of the backbone with an undecorated mannose residue in the -1 subsite. We solved the structure of the GH76. We couldn't get a complex, but we were able to superimpose the substrate from a bacillus homologue. And sure enough, you see that undecorated mannose in the -1 subsite with the O2 pointing right into the enzyme. So we were quite excited by this. But when we tested it in culture, we were hoping that we'd see a shift. We could augment the growth of the medium grower with this enzyme, but we saw no effect. So just to summarize, and we tried many other things to try to explain this differential growth phenotype using indirect methods. These include comparative genomics, bioinformatics, differential transcriptomics. We did dilution mutagenesis, enzymology, and we were left wondering, what about transport? So when we compared the SUS-C and D-like protein sequences from the MAN-PUL2, this was the only situation where we saw this partitioning of sequence with growth phenotype. So all the high growers clustered together, all the medium growers clustered together for both the C and D pair. So to test this, we actually had to develop a new method, and we partnered with our collaborators, which has turned out to be a fantastic collaboration. They're based at the Max Planck Institute for Marine Microbiology in Bremen. And what they were doing, they were taking similar approaches to developing fluorescent polysaccharides, but they had a different chemistry. Their chemistry stochastically placed the fluorophore on free hydroxyls. And this creates a randomized library so the cells can sample and select which substrates are possible to transport. Fluorophores can actually create quite a few steric hindrances for uptake, depending on their size and position. So how does this work? So we label the polysaccharide itself. We feed the polysaccharide in pure culture or mixed culture. The cell processes these substrates specifically and imports them. So you get accumulation of fluorescent products. And this gives the cells fluorescent properties that you can visualize. So this is some of the early work we did in 2019, where we're looking at our B theta control. First of all, we used DAPI just to stain DNA. We had a counter stain that was lipophilic to look at the membrane. And then when we looked in the green channel for our fluorescent polysaccharide, you can see this nice halo phenotype suggesting uptake. And when you overlay them, you get these beautiful images showing metabolically active cells. To show that this was actually driven by yeast mannan metabolism, we wanted to delete the 3 pathways involved in uptake. So this triple mutant strain was a gift from Liz Lowe at Newcastle. And sure enough, when we treated these cells with our fluorescent probe, we lost this staining, suggesting that it was responsible for the uptake. So it worked in B theta. We wanted to test it in our bovine isolates. And sure enough, we see very similar results, suggesting that we have a new fluorescent tool to actually study glycan transport. And so this slide really was the culmination of three years of work and really helped to crack the case for us. So here we're looking at our B theta control, and sure enough, we have uptake of our fluorophore at 60 minutes. But if we did a time course of this, we can actually get some rates. And here we're counting fluorescent cells. You can see around 15 minutes, this takes off and hits saturation around 2 hours. Now in comparison, the medium grower, not only did it take up fluorophore slower, it had a lower overall uptake, whereas our high grower kicked off right away and reached saturation earlier. And here we're looking at not only rates of uptake, but also total accumulation of signal, suggesting that the medium grower is much more efficient. When we looked at the supernames to understand the carbohydrate profiles, we saw a very interesting pattern as well. So in the medium growers, there's an accumulation of mannose, and also the polysaccharide itself is remaining less digested. When we quantified the mannose in the supernatant, again, we saw more in the medium grower. And some work we did with Carol Arnosti at the University of North Carolina, We were able to look at the, based on the size of the polysaccharide, we can see that in the control and also the high grower, we have a quicker diminishing substrates here compared to the medium grower, suggesting that the high growers are more efficient at processing and transporting larger polysaccharides. So there's less extracellular digestion in the medium growers. There's more free and total mannose in the medium growers, and larger polysaccharides are moved faster by HGs. So what about in complex ecosystems? So this is a sample out of a cannulated animal we have here in Lethbridge. Here we're looking at this is a food particle from the rumen. You can see the staining of cells that are attached to the food particle. When we treat it with our fluorescent yeast mannan probe, you get that nice halo stain again, showing that these are active on yeast mannan. And when we treat it with fish, so this gives us some taxonomic profiling. So this is for Bacteroides or Bacteroides phylum. You can see as well that we're showing the localization of Bacteroides cells. So we can quantify this. So we can see a total cell count, which is a little bit low for the rumen, but this, remember, these have been filtered through cheesecloth. We can get an idea of how many Bacteroides cells are present. We can also get an idea of the number that are active on yeast mannan. And then we can do a total count of Bacteroides that are active on yeast mannan. So what we found is that about 6% of the cells interact with yeast mannan, and only 3% of those are Bacteroides, suggesting that there's more diversity present in the rumen that was previously believed. Can these fluorescent conjugates, which we're calling FLAPs, can they be used to differentially label cells in live animals? So this is a collaboration with Bjorn Petrie at the University of Calgary. So what we did is we set up 3 mouse groups. We had, 4 mouse groups. In this case, we're showing mice that did not have yeast mannan in their diet. We visualized uptake of the fluorescent yeast mannan in live animals and also extracted digestive systems. And here we're seeing very little staining in both cases. In mice that did not have a yeast mannan in their diet, but were gavaged the fluorescent yeast mannan, you can now see this bolus in the intact animal, in the animal, and in the removed intestine, you can see that the signal's accumulating in the cecum, and this is at 3 hours. You can actually see the fecal pellets starting to form. When we treated mice that had yeast mannan in their diet, there was an increase in intensity of signal. And what was perhaps most interesting is that you can see there's an expansion of the community into the small intestine, suggesting that other parts of the microbiome are adapting to this substrate. Where it got quite interesting was we extracted the communities and used super resolution microscopy to look at how the cells themselves are interacting with the probe. So this is in a naive community without any fluorescence. This is a naive community where we added our flap probe, and you can see now we're seeing some non-specific binding and some interaction with cells that haven't been activated. But when we look at the communities that had been activated, there's a clear shift here. And not only are we able to visualize this prebiotic effect now, we can actually count and enumerate the cells that are active. So this gives us a new way to study prebiotic effects. When we looked at the communities themselves, we didn't really see much shift between the control mice and the mice that were fed yeast mannan. Maybe a few outliers here and there, but no real trends. So we want to take a new approach here. So we're using some cell sorting analysis to see if we can pick out the fluorescent cells from these communities. And we had a couple different populations with their different cell morphologies. So we're seeing here in the P3, these smaller Bacteroides-like, and then in the P4, we have this new cell morphology, quite large. We don't know what it is yet, but this is giving us new ways, like culture-independent ways to isolate and study metabolically active cells. So we call this enriched metagenomes, and we can now do metagenomics and metaproteomics on these communities to further study them. So moving on to another example, we were interested in looking at the digestion of polysaccharides that originate in the ocean. And this is building upon previous work. So back in 2010, there was a paper that came out in Nature called the sushi factor, where enzymes were identified in Japanese gut microbiota that had been transferred from marine bacteria. And so we've been working on applications of seaweed in livestock, not just for greenhouse gas emissions, which is quite a hot topic right now, but also to look at its digestion. So in 2012, Al Borston and Eric Martens and Jan Henrik published a paper in PNAS where they identified these two different Bacteroides species. One was specific for porphyrin and one was specific for agarose. We followed this up in 2018, where we wanted to characterize the pathway responsible for agarose metabolism. So we were able to identify a number of enzymes, including some very interesting intercellular enzymes, the transport system, the regulator. But we focused on the enzymes that we believed were responsible for depolymerization of agarose. And so just to go through this quickly, agarose is cleaved by two endoacting enzymes at the beta-1,4. This generates the neo-agroligosaccharides, as the 3,6-anhydro is at the non-reducing end. This is a specific substrate for GH117B. Cleavage of that residue results in agroligosaccharides, which is the substrate for GH2C. Its activity generates neo-agroligosaccharides again, and as you can imagine, this creates a cyclic pattern as systematically single residues are removed by the combination of these two enzymes. So we know these pathways exist in the human gut microbiome, but we're curious to study if they actually exist in livestock that's been adapted to seaweed. So consider many of the animals that are raised locally here, they've been grain-fed or grass-fed for generations, hundreds of years. So what happens now if we introduce a brand new substrate that theoretically it never would have seen before? So if they can metabolize it, the question becomes, where do these genes originate? Do they come from environmental bacteria that are able to colonize the rumen? But marine bacteria are not adapted to this environment. It's highly competitive and anaerobic. Do these bacteria persist in the rumen at low levels? But this doesn't account for extinction pressures over hundreds of generations. Or is this a dynamic transfer of genes from environmental bacteria? So this would be analogous to the sushi factor hypothesis, but this requires rapid horizontal gene transfer. So you have this nice system that we've developed to study this process, starting in the ocean, so studying the communities that digest seaweed in the environment. We're looking at cattle that are now consuming the seaweed and studying their rumen communities. We're also studying their fecal communities, and then what happens to the feces in compost in aerobic and anaerobic environments. We have one herd that we're working with here in the west coast of Canada, the Beaver Meadow Farms, where this farmer is actually very interested in scientifically studying how seaweed is consumed on farm. So what we've been able to do is look at these rumen communities, and we want to characterize the genes that are present. And one way we're doing this is extracting the seaweed cell walls and using them to generate flaps. So we're labeling the polysaccharides from this natural source directly, and then feeding them to artificial communities in Lethbridge. When we did this, we actually saw a remarkable result. So we took the communities from the cow, we adapted them to seaweed, and then we fed them with our seaweed flaps, and we actually saw uptake of this substrate and this remarkable cellular morphology. So we have these chains that are being formed, and we still actually do not know what these organisms are. We're actively pursuing this. So seaweed consuming rumen bacteria exist, and just to remind you, the seaweed that was fed to this community originated 850 miles away from where these cattle are raised, and they're landlocked, separated by the Rocky Mountains here. So we're seeing evolution of, are we seeing evolution of glycan metabolism on human timescales? So we followed this up. We wanted to look at the genetic level, so we've been shotgun metagenomics, and we've been able to identify pathways specific for seaweed, it's polysaccharides. So these GH16 subfamily 17s are They cluster with these well-characterized enzymes that are known to be kappa carrageenanases. And we're continuing this study using mag assembly, selection and cultivation of rumen isolates. So we have actually been able to isolate bacteria that do metabolize and grow on these seaweeds as a sole carbon source. We're doing metaproteomics in collaboration with Phil Pope, and we're studying the glycomics of these seaweeds and marine plants in extensive detail. So I just want to thank my team for this work. In particular, I want to point out Leanne Klassen and Dr. Greta Renges, who have been driving the FLA-PS project. Also, Jeff has been working, leading our efforts into the seaweed adaptation in ruminants. Mike is our local glycomics expert, and anyway, a great group of people to work with. I want to also thank my funders and our amazing collaborators at Max Planck Institute, University of Victoria, our local collaborators that do all the animal care and allow this animal work to be conducted. Carol Arnosti has been instrumental in helping us develop some FLA-PS methods. Bjorn, Pina, and Karen have been collaborating on the live animal imaging, cell sorting work and visualization as well. And also like to thank Mike Hahn for his work and collaboration on the Glycome profiling. So thank you for your attention.